All right, welcome. I'm going to be doing the next two modules in class, and so this is really my first introduction to you guys. So I'll tell you, I'm pretty excited about this module in particular. So I'd like you to kind of get to know me and get to know sort of where I'm, who I am, and where I'm coming from. When I was in high school in the suburbs of Chicago, and there weren't a lot of required material that had to be taught in biology at that time. And so I took a biology class, and the class was mostly just memorizing many different animals, memorizing their like land speed, their mass, their dimensions, and then sort of fun facts about all the animals. And then the next semester was plants. We did the same thing with plants. And it was really, really boring. It was awful. It was just, memorizing all these things. And I got to college and I said, well, I definitely don't want to study biology. That is the last thing I'm interested in. I'm more interested in science and things where there's like a proper kind of framework to understand it. And so I majored in math and computer science at University of Illinois, and I got to my junior year and realized that, well, I have to take a biology class. I have to take biology for non-majors in order to graduate. So I took biology to non-majors, for non-majors, and I'll just tell you that I had heard of evolution when I was younger. I came from a very conservative, religious family, and the only thing I knew about evolution was that, if you learned too much about it, you would go to hell. So not only were we not taught evolution, but I kind of actively avoided it. And I got to chapter 17 of my biology book, and that was the chapter on evolution. And I read it, and I honestly had no idea what I was going to find there. But I read it and I was blown away. I had this epiphany because like they had made me memorize like all, this entire encyclopedia on plants and animals with no framework for understanding how things were related. And then when I realized that everything is on a single tree of life, it completely blew my mind. And then, and this part sounds silly, I got to the end of the chapter and I saw a tree, and humans were also on the tree. Like we weren't separate from all of the other animals that were undergoing this gradual evolution. And that like blew my mind again. And I instantly knew it was true because I had like memorized like all these random facts, about all these plants and animals. So I took some time off school. I came back, and in my remaining year, I just did all the requirements for like a degree in general biology. And from then on, like I knew what I really wanted to do was like learn more about this field. So I was coming from a math computer science background, but I had this like new enthusiasm for learning biology. And so I eventually ended up doing a PhD with David Baker at the University of Washington, and David is well known for protein structure prediction and protein design. And so most of my graduate thesis was on the protein folding problem, and I'll talk a little bit about that today. And when I was in David's lab, gosh, we were so excited about protein structure, and I just thought bioinformatics and like, just counting those ACs, T's, and G's, that just sounds like, weird accounting and bookkeeping, and structure was the important stuff. Um, of course, I told David for my thesis, I would come up with a theory that predicted how fast a protein would fold given its structure. So if we look at the structure, some proteins fold in milliseconds, even microseconds, and some proteins take minutes to fold. So what's the difference? I would come up with a theory that explained that. I came up with a theory, and I had only been at it for like 2 or 3 years, and I said, "Hey, David, I have a theory that seems to work. Can I graduate?" And David had already gotten in trouble from the department for graduating students faster than all the other PIs. So he said, "No, you can't. You have to keep doing stuff." And so at that point, I started moonlighting at a local biotech, and that was when the Human Genome Project was just starting. And the excitement of like going into, so there were a lot of genome companies that at that point, and a reasonable business plan was you just look at the human genome, you find the genes, and then you patent them. And so if you can predict anything about the function of the genes, then you would try to, make a patent that where the use was addressing that function. And if you didn't, you patented it as an additive for toothpaste. Because there's plenty of proteins that you can add to toothpaste, just to get the patent on that gene, and maybe you could add other things later. So I started working for one of these companies, and just going through those gigantic files of A, C's, T's, and G's, and then trying to find where the genes were, and actually like looking at that at those raw text files, and seeing the ATGs and the start codons and the promoters was really exciting. And that kind of tipped me away from protein structure into genomics and bioinformatics. And I've kind of been in that space ever since. And I've tried to get students here at MIT who worked with me interested in protein structure, because that was always an interest of mine. But as of yet, I haven't. But this is kind of my first foray back into protein structure, is preparing these lectures for you guys. So I'm really happy to be here and to have the opportunity to do that. And I will say I don't actually have many slides today, and I think they may take a long time to get through. But I really encourage you guys to stop me and ask questions. This is the first time I've presented these slides, and there might be things that are missing, and so it'll be really helpful for me if you guys ask questions, and I can add that material to the slide deck for future years. So also thanks for your patience in bearing with me. All right. So today we're going to talk about what structural biology is. I'm going to walk through a case study for you guys with a transcription factor from the LAC-I family. I'll tell you how protein structures and other macromolecular structures are generally determined. I'll give you some of the basics on understanding protein structure, how to compare them, and then we'll talk a little bit about energy functions, and we'll get that connection back to physics. So in the next couple of lectures, we're going to be using deep learning to do a lot of the things that, when I was in grad school, we used to do with energy functions. But I think it is important to have that understanding of like what energy functions are. And it turns out there's a really deep relationship between deep learning and statistical mechanics, and the physics that governs small numbers of molecules. So the statistics there is very similar to the stats that we're going to use for deep learning. Okay, so let's get started. So what's structural biology? At the molecular level, structure really implies function. And I want to walk through an example where we know the function a priori, and we're going to drill down and see how the structure determines the function. So structural biology could be anything from looking at structures that someone else determined and trying to identify something about structure, maybe some commonalities between all the different enzymes that perform a specific catalysis, or it could be actually determining the structures. And there's a number of different ways that that people do that. Because a lot of the recent advancements in the deep learning of structural biology are focused on proteins. Proteins are also going to be the central focus of what we cover, and really they kind of are the main focus of structural biology. And the grand challenge for many years has been, how do we go from a protein sequence, which we can read directly off the genome, to a three-dimensional structure, if that structure helps, determine our function. And very recently, in the past, 4 or 5 years, AI methods have really started to make huge progress towards solving that problem. We currently have about 200,000 experimentally determined protein structures, and many-fold more computationally determined structures, and many of those are likely quite good. Okay, so that's sort of what structural biology is. Why do we care about it? Well, we have these genomes. We have plenty of genome sequence. I made this claim a few years ago. I don't know if it's true, but I think genome sequencing is one of the most efficient and inexpensive ways to turn a collection of matter into digital data. And we've amassed a huge corpus of genome sequence data. Much of it we don't really understand. We don't understand what the function is. So the dream is that, well, gosh, if we could convert all these sequences to structures, and structure implies function, then maybe we'll better understand what the function of these different parts of the genome are. There's a lot of diseases that result from problems with protein folding in particular. So we've talked a lot about Alzheimer's in this class, because that's a particular research interest of Professor Kellis. That's related to a misfolding of a couple of proteins, beta amyloid and tau protein. Parkinson's disease is related to misfolding of a different protein. So is Huntington's, cystic fibrosis. Prion diseases are very interesting. They're related to the misfolding of a protein called PRP, prion protein. And that misfolded protein itself is actually an infectious agent. So if you get the misfolded protein from another organism, then that can catalyze the misfolding of your native proteins. Sickle cell anemia is a genetic mutation in hemoglobin that causes hemoglobin to form large macromolecular fibers, and that can distort the shape of red blood cells. And so a little bit of distortion is protective against vectors like malaria, whereas homozygotes tend to be quite sick. And then there's an entirely new field that's sprung up around protein design. And that's the idea that, if once we've learned how to predict what the shape of a protein is, can we specify a shape that we think is going to carry out some enzymatic reaction, or maybe bind to another protein that we want to activate or inhibit? Can we design a protein that will have that structure and that function? And that's a growing field, and it's really been enhanced by deep learning methods recently. Okay, so here's some key resources that you guys are going to use. There's a number of different ways to visualize these structures. There's, you can go to the PDB website, which is called rcsb.org, and you can view them. You view the 3D structures directly on the website. But I encourage you guys to download a tool called PyMOL. PyMOL also helps to visualize these structures. How many have already used PyMOL? Oh, okay. So many of you guys. And if you haven't, it's quite useful. There's a limited set of commands that are relatively straightforward to learn. And then any command that the interpreter doesn't know, it assumes is Python. So you can generate your own fairly sophisticated code just directly in the PyMOL window. We'll talk about AlphaFold on Thursday, and that's really the deep learning approach that's had really a massive impact on the field. And then more recently, other proteins like programs like ESMFold and have had a big impact. And you can use not only the programs themselves, but there are very large databases of pre-computed structures that you guys can use. All right, so I wanna look at a case study here just to get an idea for like how do we actually how do we understand the relationship between structure and function? And most of my work is on prokaryotes, and one of the most common types of transcription factors in prokaryotes are transcription factors that have a helix turn helix motif. And probably the best known family there is the lac-I family. So what I want to do is I want to look at that protein and try to understand how it carries out its function of not only binding to DNA, but also controlling the expression of the gene that's downstream from it. Okay, so here's a view of our structure. And what you can see is you can see a protein here. So you can see the alpha helical structure. You can see the helices there. You can also see the double helix of DNA. And the first thing that we notice is that there are many copies of DNA in this picture, right? And the reason for that has to do with the fact that this is an X-ray crystal structure, and so this structure is determined by creating a crystal of the protein. And when we determine the structure, we determine the structure of the entire repeating crystalline unit. So we can kind of see how they all stack together. And in this case, there's more than one copy of the protein that we're looking for in that crystal unit. So the first thing I want to do is remove the additional copies. So I'm going to say select, I'll call it one copy, and then I'll define that as using the different chains. And so all of the different macromolecules that I'm viewing are referred to as chains. And I'm going to take chain A and chain B and chain lowercase a and lowercase b, and see what that does. Okay. So now I've highlighted just one of these non-redundant units, and you can see all the atoms are highlighted in pink here. And now I'm going to remove all the atoms that I haven't selected. So I'm going to say remove not one copy. Okay. So now I'm left with just one copy of my protein. I'm going to zoom in on it here. Okay. I'll get rid of my selection. And now we get a better view of it. Okay. I'm going to hide the DNA for a minute. So let's select polymer nucleic. And then I'm going to come over here to the bar on the side where I have a few different options. So the first one is perform an action. The next one is show, and that's going to determine what gets displayed. Next one is hide. The next is label, and the last one is color. Okay, so I'm going to choose my selection here, which is just my nucleic acid. I'm going to say hide everything. Okay. All right, so now I just have my protein. And the first thing I want to do is look at what is the structure of this protein. So when I loaded it up, I said there were two chains, right? So let's color these by chain. And so we see really the same protein repeated twice. So we're going to transcribe and express a single protein chain, and those individual chains are gonna find each other, right? And they're gonna bind to each other. And so the functional unit is a dimer, and it's a homodimer because it's an identical amino acid sequence. All right. Now I want to look at this and do a calculation. So I'm going to go to action. And I'm going to say generate vacuum electrostatics. And this is going to tell me something about what this object looks like physically. Okay. So here, the red colors indicate that if you were to bring a charge near that part of the protein, it's gonna react to a net negative charge on the surface of the protein, whereas the blue would indicate a net positive charge. Okay, so what can you tell me about this structure? Like, what are some general features? Yeah. Yeah. Did everybody hear that? So there's a positive charge. If we didn't know where the DNA was binding, we know that DNA has a very negatively charged backbone, right? It's linked by all those phosphodiester bonds. So if we wanted to have a protein that's going to orient correctly and bind to the DNA, we would want something that generally fits the structure of DNA with a lot of positive charges. And then if we had negative charges somewhere else, then that part of the protein is going to be oriented away from the DNA, right? Okay, so we can look at that and we can see if that's true. So I'm going to select polymer nucleic again. And now this is in parentheses, it says SELE. That's my most recent selection. And I'm going to go to S for show. And I'm going to show that as, let's show it as a stick diagram. and I'll color. Okay. Okay, so now we can zoom in on that. And in this stick diagram mode, we can see that negatively charged backbone of the DNA. We can see it really going directly into that positively charged cleft in the protein. And if we look at the structure of the DNA, can you guys see where there's two different qualitatively different parts of the double helix, right? There's um, there's this region in here where the backbone is quite close, and that's the minor groove of DNA. And then next to it is this region that's open, and that's the major groove of DNA. And so the major groove is where the transcription factors are going to be able to poke their residues into the DNA double helix and actually interact with what's there. So they have to kind of be able to touch those DNA residues to understand what the sequence there is. All right, so let's select chain A within 6 angstroms of our nucleic acid. And let's display those residues. So go to show. I'll use the sticks again, and I'll use the same color scheme. By element. And now I'm going to get rid of my electrostatic surface, so I can really see what's going on. So try to get a good view for you guys. So now I can look at that helix, and there's one helix here that is it's really pointing directly into the major groove, and I can kind of see these residues that are actually touching the DNA base pairs. I can see other residues like this that are touching the phosphodiester backbone of the DNA, and so those are helping to hold the protein onto the DNA, whereas these residues right in the major groove there are serving to make it specific for one particular amino acid or nucleic acid sequence. Okay, so if I had a multiple sequence alignment, then I'd expect to see a lot of conservation across different members of the family for those residues that are binding the backbone non-specifically. Whereas I might see some variation in the sequence for the residues that are determining the specificity. And so this helix in the helix turn helix motif is called the recognition helix, and it's the only one that's really crammed right into the major groove like that. When I look at the structure, it tells me something else about the function of these transcription factors. And so because it's binding to two spots on the DNA, and the vast majority of prokaryotic transcription factors are like this. They're homodimers, and they bind to two separate regions, usually separated by about 10 base pairs or so. It means that my DNA sequence is going to be palindromic. And so it's going to be the same reading from both sides as long as I take the reverse complement. So this recognition helix over here is the same as this recognition helix over here, and they're both sitting right in the major groove, right? And so one of the ways that we identify DNA binding motifs in prokaryotic genomes is by looking for conserved palindromes that are conserved across different species. All right. A few years ago, we recognized this property of helix turn helix domains. And so here you can see another zoom in of a similar structure. So this is, I believe, the pure R protein. And helix number 2, that's the one here in the major groove. And that's what we call the recognition helix. And you can see for different versions of pure R, we have this TVS motif, and these are 3 residues that are sticking directly into the major groove and determining a lot of the specificity. Oh, okay. And we were able to use this by looking at different members of this family, some of which bind the same DNA sequence, some of which bind different DNA sequences. We're able to identify 3 residues that, for the most part, determine the specificity. Now, the rest of the protein binds, for this protein, generally binds small molecules. And in the case, let's say, of pure R or lac-I, they bind carbohydrates. And so when the carbohydrates are bound, let's say it's lac-I, it binds lactose or a lactose derivative. Then it changes the conformation, so it no longer easily binds to the DNA. Okay, and so depending on the presence of lactose, this lac-I or lac repressor is either going to bind its cognate DNA or not. And what we're able to do is identify those residues. So there's many different proteins in the family, and the different proteins in the family tend to bind different substrates. So some of them bind purines, some of them bind lactose. Generally, they bind carbohydrates, but not always. And we found that if you change these 3 residues that determine specificity, you could steal the specificity from a protein, a different transcription factor, maybe even in a different species, but have it still sense the molecule that it originally sensed. And so this kind of opens the door to designing switches that respond to a whole variety of different chemicals in the cell, and targeting them to different regions of the genome. And then if we look at structure at a larger level, So we have our let's say our LAC-I protein that binds lactose and then no longer binds to the genome. If we look at how these genes are laid out on the genome itself, what we see is that the LAC-I gene controls the LAC operon. And in particular, I'm going to just highlight here. So here's the LAC operon. So LAC-Z, LAC-Y, and LAC-A are required for the import and utilization of lactose as a sugar. Here's the promoter. And so the RNA transcription machinery, RNA polymerase needs to bind to the promoter to express this, the three genes that are downstream. And generally, what we see for transcription factors that act as repressors, their binding sequence is downstream from the RNA polymerase. So the RNA polymerase binds here, but the repressor is bound in front of it or sometimes overlapping the promoter, so the RNA polymerase can't really get in. And so for repressors, they tend to just block the natural expression of the promoter. There's also activators, transcription factors that act as to enhance transcription. And what we generally see in terms of the structure of the genome is that the activators tend to lie upstream of the promoter, and oftentimes they help recruit the RNA transcription machinery. So we can have a very strong promoter where RNA polymerase will bind to it very easily, or we can have a weak promoter where RNA polymerase doesn't really bind unless there's another protein bound upstream that can kind of help it. And so there's a number of different levels that we can look at where the structure, whether it's like down to the amino acid and which amino acid is touching the base in a meaningful way, all the way up to, you know, what is the order of the genes and how do these operator sites fall with respect to the promoter. It's the structure that's really determining the function. Okay, so how do we determine structures? Well, there's a few different ways. Probably what's considered the highest quality experimental structures come from X-ray crystallography. And so there, what needs to happen is we need to grow a crystal of the protein, and sometimes it could be a complex of a protein and DNA like we just saw, or maybe a protein with a drug in its active site. If we want to see, you know, how does the drug bind there? What would we have to do to modify the drug to make it a more effective drug, to make it bind tighter? We'll take that complex and then freeze dry it and try to get it to form crystals, right? And once we have a very high quality crystal, then we put those in front of an X-ray source and it's going to generate a diffraction pattern. That diffraction pattern tells us something about the density of electrons in that crystal. And so each dot from that diffraction pattern is telling us about the electron density in its path. So from there, we're just a Fourier transform away from knowing the 3D structure of that electron density. It's a little more difficult because although we have the densities, we don't know the phases. And so there's a number of different ways that people go about trying to determine the phase, taking them from another related structure or substituting in heavy atoms and trying to calculate it that way. But that's generally the highest quality structures. And a general metric of structure quality for experimentally determined structures is the resolution. And a resolution anywhere below 3 angstroms is generally very good. And nowadays, some of the best structures have less than 1 angstrom resolution, but and anything under 3 is quite good. The next technique that's worth mentioning is NMR, so nuclear magnetic resonance. This has historically been kind of difficult to do because you first have to identify in your NMR spectrum where all the residues are and try to identify like what are the interactions between residues, but it tells you something about which residues are in contact with each other. And once you know which ones are nearby, then you can use that to infer the structure. The advantage is that you don't have to crystallize the protein. And so you can look at it at room temperature. It doesn't have to be frozen. And it's usually limited to smaller proteins. For larger proteins, we have another technique that can get us data that also doesn't require the formation of crystals, and that's cryo-EM. So there we're using an electron beam to really image these molecules in their native state. And that's often used for like very large protein complexes, like entire viruses or ribosomes. And then if we have a picture from X-ray crystallography of some of the proteins that make up the virus or some of the proteins that make up the ribosome, then we can fit those crystal structures into the general large-scale structure that we get from the cryo-electron microscopy. Okay, so we've looked at structures. I want to get into some of the basics of how we understand structures. So one way to think about the structure of a protein is as a collection of angles. And so if we think about the protein as a polymer, all the atoms are fixed, the bond lengths are fixed, and the way that a single protein can take on multiple different shapes is by rotation of these rotatable bonds. And in particular, the bonds on the backbone of the protein are the most important for determining the overall three-dimensional shape. And so here I'm showing here's a protein backbone. So we can start at the N here, moving up to the C alpha, and then the CO over here. So this is one peptide unit. And you can see there's two important bonds here. So the first one is called phi, and it's between the nitrogen and the alpha carbon. The alpha carbon is the carbon where the amino acid side chain is attached. So what is attached to the C alpha is going to be different for each different type of amino acid. In this case, there's just one carbon attached. So this is alanine. And then there's another rotatable bond between the C alpha and the final carbon. And that's called, we call that bond psi. And then there's another rotatable bond, and that's between the last carbon in the current residue and the nitrogen of the next residue, and that bond is called omega. And so if we have phi, psi, and omega for every residue in our protein, then from that we can generate a rough idea of what the protein structure is. And so the only thing we're missing are the side chains. Generally, on the inside of a protein, it's very well packed, and the side chains are aligned, so they fit together almost like puzzle pieces. And then on the outside of the protein, well, they're probably moving around, unless they're bound to another protein or to DNA or something like that. The omega bond generally doesn't change, except for in some cases, if there's a proline residue, it can change. But in practice, just a list of the phi and psi angles for all the residues in our protein is sufficient to describe the shape. And now I'm going to say something that sounds weird. We usually approximate this by saying each of these rotatable bonds, so phi and psi, has three states. And obviously, if we think about, if we had a molecular modeling kit, we would say, well, gosh, isn't there kind of a continuum of different states? Doesn't it have all the angles between 0 and 2 pi? And the answer is, well, not really. And so, if we look at this structure, where we have two large atoms, so two chlorine atoms attached to an ethane molecule with two carbons in there, we can see that the preferred configuration is to have those big atoms pointing as far away from each other as possible. And so that ends up being a lower energy state. And our molecules are going to try to spend as much of their time as they can in low energy states. And so if we look at the energy diagram, we can see in general, there's kind of three states that are low in energy. There's this one down here where they're opposite, and that's maybe the lowest. There's also a state here, and there's also a state there. Okay, so there's three. And then when our protein adopts a particular conformation, we say, oh, this is the phi psi angle, for example. Then it's going to be in one of these three wells, and which one it's in is going to depend not only on the parts of the protein that are directly bonded to that dihedral angle, but also on the rest of the protein structure. Okay. So and then within these wells, well, even in a completely folded protein, we'll have a little bit of room for those angles to wiggle around. And so when we measure how much is the protein restricted when it ends up folding to a particular conformation, it's generally the equivalent of going from about three different states to one state for each rotatable bond. Okay, so I want to give you guys a thought problem. If we start there, and I say, okay, if I have a protein with 100 amino acids, and each rotatable bond has three states, and we don't care about the side chain conformations. How many total conformations does the protein have? So maybe just take like 1 minute and see if you can come up with a number. 3 to the oh wow, that's a good answer. Yes. Yes, I would have taken 3 to the 100 as well. Why 3 to the 99? Yeah, so the ones at the end don't count. Okay, but I will say you're almost right. There's one minor factor that you left out. Can you think what that is? You got the second order correction, but you missed the the what? Yeah, there's 2 rotatable bonds per residue, so it's 3 to the 200, or let's say 198. Excellent. Okay. So that's a lot, right? And if we say that 3 squared is roughly 10, then that's about 10 to the 100 power. That's how many conformations we have. How many of those conformations correspond to the folded state of a protein? Well, let's think about the shape of a protein, and if we were to take just pick any rotatable bond in the middle of it and switch it from one of those configurations to one of the other two, and so rotate it around that bond 120 degrees, that's not going to be folded anymore, right? Pretty much any bond that's not right at the end of the protein, if we rotate it to a different configuration, it's not going to be folded. So we can really say that the probability of finding the protein in a folded state by chance for a 100 amino acid protein is something like 1 in 10 to the 100 power, and this is the basis for the protein folding problem, right? And so in the 1950s, Christian Amundsen posited that the conformation that a protein adopts is its global energy minimum, its global free energy minimum. Okay, and then in 1969, Cyrus Leventhal said, "Well, that's all the evidence seems to suggest that that's true. It is true in probably most cases. Maybe the notable exception being the prion protein. The prion protein is a even lower energy state, and a lot of these misfolded proteins are lower energy states. But if our protein has to sample these different conformations so it can find the global energy minimum, there's not enough time. Like even if the protein had started at the beginning of the universe, like it wouldn't be able to sample 10 to the 100 conformations by now. And yet some proteins fold in milliseconds or even microseconds. So that became the Leventhal paradox. So what do you guys, how would you guys explain that? Oh, I thought I was going crazy. I thought it was like getting darker and darker. So what do you think is going on? Like these proteins have to find the one conformation that is lowest in energy, and there's so many to search through. What's the like? What do you think is going on? And if I can paraphrase that like a little bit, if we looked at our protein structure and we calculated its energy and we said, look, we can turn some of these rotatable bonds now, and we used an algorithm like gradient descent, right? Where we said, okay, if I turn this bond a little bit, is the energy going to go up or down? We would probably find that there's substructures on this protein chain where if I make a move there, it's lowering my energy. And the first thing that's going to happen is these local interactions between residues that are adjacent or nearby in the structure. So in 1998, this graphic came out that I think was explained a lot of how we get over the paradox of protein folding, and that says that the contact order. So on average, how far apart two residues are that contact each other, the farther apart they are, the slower the folding. And on average, if most of the contacts in the protein are quite close together, then the protein can fold quite rapidly. Okay. And so that brings us to like the different levels of protein structure. So if our primary structure is just our chemical structure and our linear amino acid sequence, the first thing to organize itself is then interactions between these residues that are adjacent. And so that's the secondary structure. The tertiary structure I'll define as just the global conformation of our 3D protein chain. And then like our LAC-I example, where we have two different protein chains coming together to make a higher order structure, that's our quaternary structure. And so I want to familiarize you guys with these plots. So this is a Ramachandran plot. And this is constructed by looking at many different proteins, many different amino acid residues within those proteins, and then looking at their phi and psi angles. And you can see that not all phi and psi angles are accessible. There's a really dense region here, and phi psi angles in this regime tend to form alpha helices, whereas phi psi angles here tend to form beta sheets. Phi psi angles over here, I think, are usually restricted to glycine. So glycine doesn't have a side chain and it can adopt lots of different shapes. And then the rest of them tend to occur in more disordered regions of the protein or turns that connect, alpha helices and beta sheets. So if you look at alpha helices and beta sheets, we can see one of the reasons why they're so favorable and is that they have favorable stabilizing interactions. And so here's our beta sheet, and you can see all of these bonds here. These are hydrogen bonds. And in our alpha helix, we have hydrogen bonds too, kind of propagating down the direction of the helix, whereas in the beta sheet, they're between adjacent strands of the beta sheet. And these beta strands can be going the opposite direction like this. So here we have this guy going to the left and next to the right and then back to the left. So they can be anti-parallel, or they can loop around and come back in the same direction and form parallel beta sheets. There's been a bunch of different approaches to predicting protein secondary structure. The first big one was the Chow-Fastman approach, and for that, they really used a statistical approach. So looking at different amino acids in crystallized protein structures, and then computing three numbers: the probability of observing that amino acid in an alpha helix, observing that amino acid in a beta sheet, or observing that amino acid in a random coil, or just anything besides alpha and beta. And then looking for stretches along the protein sequence of amino acids that seem to have a preference for, let's say, helix or beta sheet. In 1993, another program really took over and made much better predictions of secondary structures, and that was really one of the first times I had heard of neural nets in biology. And so that was just a three-layer neural net, but it also incorporated data from multiple sequence alignments. So it also looked to see if homologous proteins also had amino acids that were consistent, and either in helices or beta sheets. Nowadays, protein structure prediction, three-dimensional protein structure prediction is actually quite good. And I think to some extent it's kind of circumvented predicting secondary structure. So for tertiary structure prediction, we want to go from a full linear amino acid sequence all the way to a three-dimensional structure. And one of the key steps in that is identifying a protein contact map. And so if I just plot out all of the residues in my protein on both the x-axis and the y-axis, I'll see my alpha helices right down the y equals x line, because they tend to interact with other residues, about 3 steps ahead or behind them, and make hydrogen bonds that way. And these linear stretches are parallel beta sheets, and the ones that go perpendicular to the x equals y line are those anti-parallel beta sheets. So the same kind that we saw in the previous diagram. This observation that we can go from protein contact maps to 3-dimensional structures relatively straightforwardly was the basis for a lot of the methods that use coevolutionary information. So if we look at a multiple sequence alignment and we see, well, gosh, whenever this amino acid changes on the tree of life, we also see a corresponding change in this other amino acid, then we can infer there's some sort of relationship between them, and oftentimes they're interacting and they're physically close to each other. So one of the early approaches to structure prediction was to incorporate those coevolutionary associations as contacts, and then try to infer protein structures that had contact maps that looked like the coevolutionary maps. In a couple of lectures, we're going to talk about protein language models, and we're going to look at residues in the protein language model that the model is telling us have a lot of interaction with, or need to pass information back and forth between other residues in that protein. And that's done through something called attention maps, and we'll see maps that look very similar to this. Okay. Protein structure prediction milestones. This really started when there was a competition to see, how well protein structures could be predicted. So for a while, there were many papers claiming to have solved the protein folding problem. And then what they did is they got some crystallographers together and they said, what structures are you all working on? And then they released the amino acid sequence of the structures that were, currently being determined, but had not been published yet. And they let the predictors, use their methods to predict the structures. And so for many years, very little progress was made, and it turned out that nobody really had a very good way of even getting like small regions of totally novel proteins predicting their structure. While I was a graduate student in David Baker's lab, he developed a program called Rosetta, and the unique aspect of Rosetta compared to some other programs is, although it was sort of physics-based, it used statistical tendencies of different stretches of amino acids to fall into different regions of that Ramachandran plot and have different phi-psi angles. So you would actually build up structures by mixing and matching short stretches of DNA and just taking the phi and psi angles from other protein structures, like maybe 5 amino acids at a time or something like that, and just stringing them together, and then estimating like which ones formed nice compact shapes using an energy function. And so at the time, that made predictions that kind of surprised everyone, although that for most proteins, they probably wouldn't be considered good today. And then in 2020, AlphaFold 2 came out and really started to generate structures that were a huge leap from what people had seen before. And these were using deep learning methods together with evolutionary information from multiple sequence alignments. And the newest methods are nearly as good and don't require multiple sequence alignments, so they can operate on a single amino acid structure. Okay, so once we have a protein structure, so here's an example of a protein structure. This is obviously like a very good prediction, but in the blue is a structure that was predicted by AlphaFold. In green is an experimentally determined structure, and you can see they match quite closely. But on in general and on your homework, we want to be able to calculate a number that specifies like how good is this match. So there's a few steps involved in doing that. First, we have to find all the corresponding residues. So if we're looking at two different homologous proteins, we need to do a multiple sequence alignment and identify those corresponding residues. If we're comparing a predicted protein to an experimentally determined protein, then the mapping is very straightforward. And we can either try to map every single atom, or we can say, let's just take one atom from each residue. Let's say the C alpha carbon is often used for that. So the parameter that's usually, or the statistic that's usually used to compare structures is the RMSD, so the root mean square difference. And it's exactly what it sounds like. So that's that distance is just if we look at our x coordinate of protein 1 and our x coordinate of protein 2, take the difference, square it, do the same for the y and z coordinates. Then that becomes our distance, and then we just add those distances, divide by the total number of residues that went into the calculations. So we have our mean distance, mean squared distance, and then take a square root. So relatively straightforward. What we need to do first, though, is we have to have the two proteins properly rotated and on top of each other. So we can compute the center of mass of each protein. And for that, we just take the average vector to each of the atoms. We add them together, take the average, that's our centroid. And then we're just going to subtract that number from each vector in that protein. So we're going to shift the protein down to the origin. But then we have another problem. And we have two proteins that are at the origin. They might be really, really similar, but one might be rotated 180 degrees. So the root squared difference between those structures is going to be large. And it turns out that if we just compute after they're centered at the origin, if we compute the covariance matrix, from that we can do a singular valued decomposition and get a rotation matrix that we can then apply to either one or the other of the protein structures. And that we know will give us the optimal superposition, and then we can compute the root mean square difference between those. And the last thing I want to say about some of these approaches to analyzing protein structures is the energy functions. And so probably next day I'll finish talking about that, but I'll introduce it today. So all of our physics-based methods, and certainly all the early methods in protein structure prediction, made use of these energy functions. And the energy functions try to approximate the physics of proteins as best they can. And we can't get the exact energy functions because a lot of what's happening in proteins is very strongly quantum mechanical, and so the calculations are just beyond what we can do. But most of these functions are parameterized based on the statistics of like many protein structures to be able to perform certain tasks like protein design or protein structure prediction, docking of two proteins together, or if we're doing drug design, docking a drug into the active site of a protein, or taking an existing drug that binds to the active site of a protein and identifying like what are the changes that we would have to make to this drug molecule to make it bind tighter, for example. Molecular dynamics kind of takes those energy functions and integrates them forward in time, so we can see, you know, what would this protein look like if it was in motion. We can use them to predict the effect of mutations or even design de novo proteins. So next day we'll look at a few different common components of energy functions. Electrostatics, so attraction or repulsion of electric charges, van der Waals interactions, so kind of steric occlusion of different residues. We'll look at hydrogen bonds, and then the hydrophobic effect as well. So I will stop there.